Hello, my name is Anika Liu and I'm a PhD student at the University of Cambridge and funded by GSK. Today I'd like to present our recent work studying which cellular changes take place before drug-induced liver injury, or rather the earliest histopathological evidence of it. The underlying assumption thereby is that if certain cellular changes consistently take place before adverse histopathology, these are time concordant and potentially causally involved in the pathogenesis. And we believe that this kind of time concordance analysis is yet underexplored and could, for example, also be applicable to other mechanistic studies on other safety or efficacy-related endpoints. So how do we study time concordance between cellular and adverse event? We use data from repeat dose studies with daily dosing across 170 compounds at multiple doses from the TG-Gates database. And in these studies, measurements are taken at 8 different time points, out of which 4 are within the first 24 hours and 4 are between 3 days and 4 weeks. These measurements include gene expression in histopathology in the liver, and we use the gene expression to derive pathway and transcription factor activation as cellular events, while adverse histopathology indicates organ-level injury, so the adverse event. For each of these events, we then derive the earliest time point of activation. So for pathways and transcription factors, this is the first time point with significant differential activity in comparison to the vehicle control. Here it should be added that the behavior after this first activation is not included in the time concordance analysis, as we argue that, for example, both temporarily as well as continuously activated events can be mechanistically important. Of course, our confidence and time concordance from one individual time series with 8 time points is not very high. However, we have a large set of time series and can therefore test whether the event frequency before adverse histopathology is significantly higher than expected based on the background frequency observed in non-adverse conditions. So with this analysis, we are able to identify time-concordant pathways and transcription factors, which are significantly enriched before adverse histopathology. We then thought maybe we can not only classify time-concordant or not, but also prioritize the most promising events looking at the temporal relation. Therefore, we introduced a set of nice-to-have properties on top of the significant enrichment, and these indeed identify known and plausible events in DILI. We first looked into the most significantly enriched events and find downregulation of bile acid and glycosaminoglycan metabolism. Looking closer into why these were most significant, we find that they are never observed in non-adverse conditions, so have a high positive predictive value, but are still at the same time relatively frequently observed before adverse histopathology. However, there were more frequent gene sets found, with the most frequent ones pointing to changes related to ER stress mediated by eukaryotic initiation factor 2 alpha. These are less significantly enriched due to the lower specificity, which aligns with the role in adaptive response which precedes DILI but is reversible. In addition, we found pathways related to lipid metabolism with the largest effect size before adverse histopathology, so these may not be the most specific, frequent, or significant, but if they are found, they change strongly and are hence potentially easy to detect experimentally. Of course, a lot is already known about causal interactions in biology, and we also wanted to take advantage of this prior biological knowledge. So at this point, we have included 3 kinds of interactions. The first one are transcription factor target gene interactions, so knowledge on which transcription factor controls which gene, which is also how we inferred transcription factor activity in the first place. So this was done with Dorothea, which I've referenced below. And the second kind of interaction is the central dogma of molecular biology, or more precisely, that transcription is followed by translation, which means that in case of transcriptional regulation, we should see changes in transcription factor expression before we see changes in the activity on the protein level. However, of course, transcription factors can also be activated post-transcriptionally, and here we additionally include protein-protein interaction as a third source of prior knowledge, which can shed light on how transcription factors regulate each other on the protein levels. So among all the identified transcription factors enriched before adverse histopathology, we then again look for time concordance between transcription factor and gene level events, which are also supported by known interactions with the matching sign and direction. What I've shown here is the largest cluster identified in this way, and this provides detailed hypotheses on cellular changes in DILI. One interesting case is the interaction between CEBPA and HNF4A, which I've circled here. Interactions between both are known in both directions based on prior knowledge. However, one direction is more frequently observed to be supported by time concordance, which is the darker line here. So in this case, time concordance is able to prioritize one direction, which would not have been possible otherwise. In summary, we believe that time concordance is a useful way to identify mechanistic hypotheses from time series data. We demonstrate its utility using transcriptomics and histopathology data in the context of drug-induced liver injury, but would also like to highlight that this can, in theory, be applied to other types of data, for example, other omics or data on other timescales. Last but not least, I'd like to acknowledge my supervisors, Andreas Bender, Namshek Khan, and Jordi Munoz-Meriadas, as well as the Bender and Hahn groups. Also, if you have questions, feedback, or suggestions on this work, please get in touch.